Novel therapeutic inventions that address the cause of age-related damage and dysfunction hold great potential to prevent or treat age-related diseases and extend the healthy human lifespan. In this session, company builders and senior leaders of emerging organizations share the latest approaches in tackling age-related disorders as the next frontier. This session features Rita Ballas-Gordon, CEO, Muna Therapeutics, Marco Boersma, General Partner at Forbion, Jens Eckstein, Managing Partner, Apollo Health Ventures, Kristen Fortney, Co-founder and CEO, BioAge, James Pyre, Founder and CEO, Cambrian Biopharma, and Session Leader Dave Madge, VP of Discovery Services, WuXi AppTec.
Hi everyone, and welcome to the penultimate session in this Healthy Aging Forum. My name is Dave Madge and I work in the Discovery Services team at WuXi AppTec. In this session, we'd like to build on the insightful contributions from previous speakers and panelists and really start to look at what the future may hold, how we're going to realize real advances towards improved health span. Over the next 40 minutes or so, we're going to discuss with key leaders from diverse fields of healthy aging, investments, research, and development. So please let me start by introducing my guests on this panel. Rita Ballas-Gordon, CEO of Muna Therapeutics. Hi, Rita. Kristen Fortney, co-founder and CEO of BioAge. Hi, Kristen. Marco Boersma, general partner at Forbion. Jens Eckstein, managing partner at Apollo Healthcare Ventures. And James Pyre, founder and CEO of Cambrian Biopharma. Thank you all for joining us today. So to get us started, I think we're all familiar with the basic tenets of the interwoven hallmarks of aging, considerations of genomic instability, cellular senescence, stem cell exhaustion, and other actors. James, I think you would categorize these elements very broadly into molecular alterations, cellular dysfunction, and tissue degeneration. Amongst these broad categories, what are the disease areas that you feel might prove more accessible to therapeutic intervention in the near term?
I think one of the fascinating things about this ecosystem, using the breakthroughs that we've made in science all around the world into understanding what causes aging, and I think represented by many of the folks that we have here on the panel, is that the number of diseases that are going to be first looked at by drugs coming from this longevity field, broadly defined, are incredibly diverse. Everything from, chronic diseases like arthritis, diabetes, neurodegenerative disease, to acute age-related conditions like cancer or myocardial infarction, but even things that kind of even come out of left field, like pediatric rare childhood disease or pediatric rare diseases that share a mechanism of action that has gone completely haywire with something that normally degrades bit by bit as we get older. And so one of the things that I always like to think about as we go into a discussion like this around healthy aging and healthy longevity is to make two quick points on what this field is and how to broadly define it, right? The first piece is that I think everyone playing in this field today is focused not so much on indications as they are on mechanisms. And as a field, we're interested in mechanisms of action that, I'm going to say get worse in the broadest possible terms as we age. Sometimes that means it's a mechanism that isn't there when we're young and appears when we're old. Sometimes it's something that is there when we're young and disappears when we're old. And this field really got kicked off, about 8 years ago, when a whole bunch of feuding scientists came together to identify the 9 hallmarks of aging. We extended those at Cambrian to about 13 different molecular drivers of aging, but each one of those can be addressed by new therapeutics that have been created in just like the last 3 or 4 years. And that's giving rise to a whole bunch of different indications that are targeted in these stepping stones that the industry is going to work at. And this is kind of the second point, that each drug created by an entity in this space will first be used against an acute condition, something that's regulatable by the FDA or another regulator today to show safety and efficacy. And then after you have a safe and effective drug targeting one of these drivers of aging, then you can say, okay, now let's use this in a prevention context, in the same way that, you know, statins, for example, were developed to extend human health in a primary prevention way in the long term. So I think it's a long answer to your question, but I think frames a little bit of the space that we're playing in and the incredible diversity that you're going to see from these novel mechanistic breakthroughs applied to all different areas of human health.
That's very, very helpful, James. And as you say, provides a great framework for some of our further discussion. And perhaps this is a good point to have a chat with Rita and learn a little bit about how Rita's focused on developing therapies for neurodegenerative disorders. You know, this notion of building on insights into molecular pathways in different brain cells, different cell types that underlie disease pathology is obviously going to take us in different directions. Different therapeutic opportunities will arise in different CNS disorders. Rita, you in particular, you're identifying pathways whose manipulation can lead to resilience to the development of neurodegeneration. These are obviously complex pathways, so how do you build that understanding between disease progression and a tractable point of intervention?
So much of the field of neuroscience over the last 3 decades has been focused on the mechanisms that drive neurodegeneration and what the sequelae of those mechanisms are for brain function, notably cognition. And we and others in the field over the last 5 years or so have taken that information and sort of turned it on its head and asked the question, what are the mechanisms that lead individuals to age very successfully and retain cognition at a very high level despite the presence of hallmarks of neurodegeneration? And can we take an understanding from the cells and the tissues from those patients post-mortem and map those onto cellular mechanisms that confer resilience and are the substrate of what we think will be healthy brain aging? At Muna, we are using autopsy samples from individuals with prominent AD pathology and cognitive impairment compared to age-matched controls, and using a spatial transcriptomic approach to understand the response of different cell types in the brain surrounding AD hallmark pathology and at a distance from it. And I think that perhaps is not so surprising. I would call that a rather conventional approach. But we've mapped onto that an understanding of what cell types are doing in individuals who have prominent AD pathology, but who have been deeply phenotyped and are cognitively normal and have a putative resilience phenotype, resilience to the effects of AD pathology. And then to address the question of whether or not such individuals would develop the hallmarks of cognitive impairment and other sequelae that are mapped to Alzheimer's disease if they live long enough, we've included in these analyses the brains of individuals who are centenarians with and without AD pathology and with and without cognitive impairment. And so we have a very broad data set of individual cellular responses and pathways that are dysregulated in disease, how those pathways are affected in resilient individuals, and then how those pathways are represented in a centenarian cohort. Now, you asked the question of what do you do with that information to lead you to therapeutics? And of course, this is the issue that we and many other companies and academic labs around the globe are wrestling with. And there are a number of tools that we're taking advantage of, not the least of which is machine learning and AI approaches to really get to a bioinformatic understanding of these data sets and understand what is changing spatially and temporally in the context of disease or resilience or centenarian brains. We're understanding the biology of these pathways in human iPS-derived cells, and we're using all of the cell types that are represented in the brain. And then are asking questions by pushing and pulling on the biology for interesting nodes in those pathways by overexpression or knockdown, what the effect is of those pathways on driving cells to either a disease phenotype, a neurodegeneration promoting phenotype, a resilience or resilience promoting phenotype. And of course, lots of other filters on the data set, including focusing on those pathways and nodes where there's some existing human genetic evidence, for example, or an understanding of some of the core biology. And then putting all of this evidence together, the idea is that this approach will lead us to some familiar targets. And I think we can say, although it's very early days for Muna's work, that some of those familiar targets have already popped up.
Thanks, Rita. You speak very, very deeply about new technologies that are contributing to our ability to identify targets and to make disease associations in this space. And I think this leads very nicely into perhaps a discussion from Kristen as to what bioage are doing, which is, again, very much enabled by new technologies and building a sort of bottom-up, ground-up approach to understanding what's happening in different cell types in aging. So, Kristen, could you talk to us a little bit about how you're forging an understanding of the aging process and how you're identifying targets that can be beneficially manipulated? Perhaps in particular, what are the mechanistic insights that led to the selection of your current clinical portfolio?
Yeah, lots of great questions there. So definitely, so at bioage, yeah, we're clinical stage now, and we're really focused on building out a portfolio of differentiated drugs that address different mechanisms of aging. And the targets for these drugs are really emerging from our human data platform. So I think, like everybody on this call, I'm very bullish about the aging space. I think there are a lot of things that are going to be discovered. So at bioage, we are working with thousands of samples, thousands of human individuals, where there are samples collected from some really special biobanks that started collecting these blood samples and health records and health variables as long as 50 years ago when these people were middle-aged. And those individuals were tracked longitudinally for the entire rest of their lives. So this is really rich data. And I think that if you really want to get a handle on human aging, you need these kind of data because humans age on the scale of decades. And ideally, you want to know what's going on pre-disease, right? Because these deleterious aging changes that predispose to disease happen before you become very sick. So our approach at bioage is really to go in and into these samples and use these modern technologies like the proteome, the metabolome, the transcriptome, just list out every molecule that we can. And it's a really important point that the scale of that, but what we've been able to do there is very different from even just a few years ago, right? Like if you're doing just a proteome on the blood, you can now look at 7,000 proteins if you're using SomaLogic, and you could see maybe like 10-fold less just a few years ago. So the scale of what we can do now is really amazing. But we can make a list of all these different targets that matter and then ask a bunch of questions of this rich data set, like what is changing with age? But the most important question, I think, for us at bioage is what predicts the future, right? Like what molecular signatures differentiate those people who go on to live 90 plus and their brains still work and their bodies still work, the centenarians, right, with this compressed morbidity, from the rest of us, or people who maybe only make it to age 60. So when you have this sort of rich distribution and of all these human experiments, you can figure out these different pathways that are important for aging. And as you might expect, there are lots, right? So I think there's going to be a lot of therapies that really make a significant impact on human health span and lifespan. And at bioage, thus far, we've made 3 clinical stage bets based on some of these data sets. So just to give you an example, you asked about our current clinical programs today, and I'll just give you a brief summary of each of them. So our most advanced program right now is a drug that impacts immune aging. And this drug seems to be, not really care about the particular pathogen challenge because it's correcting certain aspects of the immune system. And this drug is currently in phase 2 trials for an older hospitalized COVID-19 population. We have 2 drugs as well that are focused on muscle aging. So one drug that we brought in really recently from Amgen, it's an in-licensed drug. It's a really exciting target called apelin. When we use this this molecule in multiple different models of mouse muscle atrophy and regeneration, it significantly improved what we saw there. So again, we're going into an acute muscle indication first, but ultimately it could be a frailty drug. And just really briefly, our third program is a hypoxia stimulator. So this is the pathway in your body that responds to low oxygen levels. Again, similar story, lifetime association with future good outcomes, interventional studies showing it can improve regeneration and muscle function. And our first indication there is going after hip fracture, post-operative anemia.
Fantastic, Kristen, thanks very much. So I guess there's a couple of themes emerging here perhaps around the enablement piece, technologies, access to substantial, well-characterized longitudinal data sets, access to human stem cells, human samples that allow us to really interrogate what's happening over a period of time. I'm interested to get a view from our investors on the panel. This is a huge amount of exciting science that's going on in the area. What are the features of new discoveries that convince you to nurture or invest in these ideas? Perhaps, Jens, you could start. What's really excited you in this area recently?
Yeah, I mean, great introductions from the three previous speakers because I can kind of skate on them. I mean, as James mentioned in the beginning, what really has changed over the last 5 years is that we have boiled down gerosciences now really to real known molecular targets. And that really allows us to use the whole armamentarium of pharmaceutical drug development, but put them on a completely different set of targets with a very, very different approach to disease. And that is, of course, good for investors because we know what pharmaceutical development can do. If we understand the target, if we understand the pathway, we can find chemical matter or antibodies or any other modality in real time, and we can actually then use those compounds as tool compounds initially and then interrogate the biology and see if the hypothesis was really true. And that's a known path. So that's great for an investor because it really mitigates the risk to go into something completely novel. So that's fantastic. So we have real targets, which is great. The second one I would say is what Kristen was alluding to, and that is the tools we have today are just amazing. I mean, if you look at tools development of the last 5-6 years, sequencing, single cell RNA-seq, proteomics, where you look at 10-20,000 proteins at the same time, metabolomics. All these have come down in cost tremendously. So this is also now something you can do in real time. So you can do an early stage program and you can pull in all these omics tools and really get incredible information. If we combine this now with longitudinal data sets, this actually grows even more. So you can have a very nice interplay between creating chemical matter that is targeting specific targets, using the chemical matter, interrogating the biology again, get feedback from the biology, and then test it against the data sets of the omics and see if this all fits together. And ultimately, you will now end up with a mechanism-based approach. So you can follow your mechanism of action from the early stage, which is out of cells, biochemical approaches into the cell-based assays, into the animal models, and ultimately into man. And that is really where the risk mitigation lies for investors. We want to follow mechanisms. We don't want to be surprised in a phase 1b or phase 2 trial where we suddenly see some things that are completely off mechanism. So that is incredibly encouraging.
Thanks, Jens. Marco, would you like to step in here with some comments too?
As an investor, you of course always look in, okay, what problem are we gonna solve, right? And healthy aging and longevity is of course very exciting and very relevant. Obviously, also with the population getting older in the first place. It's always a question. So, what is it that make people sick, and what is the disease, and how are we gonna cure those? We're not gonna give persons of 30 or 40 years of age a pill to see how much longer that person is gonna live versus persons that are not getting medication, right? So how it's currently is, and I hope it's gonna change. But for now, it's mainly, okay, what disease are we gonna treat and drug, and see if we can cure. And then ideally, of course, we get into a prevention setting rather than treatment, because I think there is so much more to gain.
Well, Marco, you recently launched Oxytope with a focus on oxidative stress. What did you find particularly appealing about the underlying science in that company?
Yeah, that's an interesting technology that's been developed. Well, that's based on 30 years of basic research by Joe Whitstone and Sam Chimikas of University of San Diego. And then you see actually the importance of basic science, and that it takes typically a while before that basic science is going to be applied in like true drug development. Oxytope is about development of antibodies against oxidation-specific epitopes. As we age, certain individuals as we age, there's excessive oxidative stress leading to inflammation, and in certain persons, in certain diseases, that is more excessive than in others. Oxidation-specific epitopes or modification of proteins and lipids generated in organs and tissues during oxidative stress. In evolution, the innate immune system has generated natural antibodies to bind and inactivate those oxidation-specific epitopes to protect actually humans from disease or from oxidative stress, but then from inflammation and inflammatory diseases. So many individuals are born with reduced levels of those protective antibodies, leading to early disease and a lack of lifelong protection. So Oxytope has actually identified those antibodies, clone them, humanize them, and the idea is to develop those initially in diseases that we all know, like atherosclerosis, liver diseases, neurological inflammations, and that is what we are doing. So we are gearing up to get this into the clinic in the next 2 years or so. Those antibodies.
Fantastic. Thanks very much, Marco, and good luck with that. So we've all spoken about some of the basic science and how this is being applied today, but I'd like to ask the panel what new basic science and technological breakthroughs might accelerate our understanding of the pathophysiology of aging and the ways in which we can intervene. So what tools would you like to have that are not available now? How critical will development of new biosensor and biomarker technologies, for example, be to further progression in this area? James, would you like to start?
Sure, happy to jump in. So I think we almost have to kind of break this topic into two, because the field faces two such dramatically different challenges, right? The first challenge that we've mostly been talking about today is, can we take these drugs that have the potential to be used as preventatives, that are kind of multi-disease preventatives to extend healthy lifespan or slow aging, and can we get those drugs approved, shown to be safe and effective in some disease? The second challenge that I think you're alluding to here a little bit, Dave, is once you have those safe and effective drugs, how do you make that jump from treatment to prevention? Because that's, I think, why we're all in this field, is that jump to prevention. The first one is that you can't easily run a multi-disease prevention trial, right? It's just long and expensive, but it is possible. You just need to have a lot of long-term investment, a lot of long-term investment to run this trial, and you need a safe and effective drug that isn't going to trip over its feet on some early endpoint tied deeply to mechanism, like Jens was saying. Like, you really need to understand the mechanism and what goes, something is going wrong in humans. And so you can run these trials, but they take a lot. The second challenge is that there's a shortcut to running those really long trials, which is to use a biomarker-based surrogate endpoint. And we don't have a suitable surrogate endpoint in this space to run those kind of trials today, but we're not that far. But such a surrogate endpoint isn't that far in the future, especially if we start doing a whole lot of placebo-controlled clinical trials with all of these drugs that are bubbling up in this field, and we're very careful about collecting data, placebo-controlled data from all of these spaces. I think Kristen's work at BioAge is a great example of this, where they've started pulling this data forward and are starting to run trials based on these interventions. And at Cambrian, we're doing exactly the same thing. That as we send programs into the clinic, we are, doing trials on existing diseases, but pulling down all of this data that could be putative biomarkers of, I call it multi-morbidity risk, as opposed to biomarkers of aging, for every type of data set that has been proposed to be one of those biomarkers. And I think the field will narrow in on a surrogate biomarker, not next year, but also not too far in the future.
Rita, this must represent a challenge for you with a CNS focus as well. I'm sure you put a lot of thought into how you might be able to manage clinical studies with better biomarkers, for example, in the future.
Yeah, I think there are two really big issues when thinking about neurologic diseases that play out over many decades. The first one is biomarkers that define the run-up to the frank manifestation of disease. So the prodromal phase, where brain function is no longer optimal, pathology has begun to develop, but it falls just short of frank clinical manifestations. There's, I think, general unanimity that intervening in the prodrome would be optimal for reducing the manifestations of pathology on brain function and perhaps even delaying them. But we have a very poor understanding of what is the prodrome, when does it begin, how does it change over time? And I want to contrast that to how much our understanding of actual disease manifestation has exploded in the last decade. So we're currently in an era where we can integrate across the multiome, as Christian mentioned earlier, not just the transcriptome, but the proteome, the metabolome, the lipidome, et cetera, and really understand how we can get our hands on biomarkers of disease and disease progression, and I guess the holy grail being biomarkers of therapeutic efficacy, right? But biomarkers that define the prodrome and an optimal window for intervention, I view as really the next frontier in neurologic diseases, and frankly, many others as well. And one that would, if we had those in hand, would greatly accelerate our ability to develop the right interventions for the right patients at the right time. The second point I'd like to make is that we have a really good understanding, and I think data sets at our disposal to integrate someone's polygenic risk score for disease, whether it's diseases of the brain or cardiovascular system or musculoskeletal system, with how their disease plays out in their lifetime. But what we don't really understand yet is how factors that are lifestyle factors, right? So what you eat, how much you exercise, how fit you keep your brain, how those impact polygenic risk score and the manifestation of disease in individuals. And I think once we have an understanding of that, we'll be in the era I think we all want to be, right? Which is having a number of simple, sort of in the cupboard or in your home personal interventions that you can do, given your particular genetic makeup, to stave off the effects of disease, and then a host of pharmacological agents that will carry you the rest of the way to either abrogate the onset of pathology or mitigate its effects. I think that's a much longer term horizon, but I think all of us would like to be actively working in this field to make sure that we get there in a timely way.
Fantastic. Kristen, I'd like to come back to you a little bit and talk a bit more about how you're using longitudinal data sets and deep phenotyping to understand a little bit more about where the disease targets might lie. And ask whether you think there might be further opportunities for the sort of quick wins that we might consider metformin to be, for example. Do you think there are going to be some near-term success stories that we can look forward to over the coming years that really validate the kind of approach that you and others are taking in this area of really understanding disease phenotype and linking that to pathways and targets, some of which may already have existing molecules.
Yeah, I definitely feel there are going to be a lot of quick wins in the field. Again, just because so few things have been tried, right? And humans, like any other animal, have not been optimized for longevity, like in any way. If we learned anything from a decade of, invertebrate aging research, it's that there's actually, this is really where the first sort of wave of exciting new discoveries came, right? Back in the early 1990s, when Cynthia Kenyon, Gary Ruvkun showed you could delete one gene and double a worm's lifespan. Wow, right? But over the next few years, what scientists discovered was that you could actually, hundreds of genes, if you modified them to upregulate them or downregulate them, also extended lifespan. So it's not just one thing, it's going to be a lot of different mechanisms. And so I think there are going to be a lot of complementary pathways that you want to hit that together can be very additive. So at BioAge, and this is what you're alluding to, I think, we're focusing first on those easiest wins, right? So from our human data sets, we have discovered all these different pathways that matter for aging. And the question for us is, which ones do we bring to the clinic first? And that's an easy answer. It's the ones that are the closest to translation, the ones that are closest to patients. And there are pathways that emerge from our data where there was already a molecule out there that's been in a phase 1 study that's known to hit its target. We can go immediately into an acute indication, a phase 2 study, and get that real de-risking human data, right? Like the sort of the ultimate, importantly too, not just whether it works for that acute disease, but also the biomarkers of aging, which I think are a really critical component. Like ideally, this therapy isn't just relevant only to this one disease, but is fundamentally changing the way you age. And then building from there.
Jens or Marco, would you like to comment on these kind of near-term opportunities that might be appealing to the investment community as well?
Yeah, sure. And I mean, you mentioned metformin, and I think it's going to be great to see what Near Barzilai's big TAME trial will bring us. But, the amazing thing is that it's really a spectrum, right? I mean, people, when you start talking with people about age-related disease, they say, well, it's too complicated, it's too complex. You will have to have so many different approaches to actually do something in combinations. And there will be combinations, there's no doubt about it. But we also know that single molecules already have an impact. And we're involved with one company that's working rapamycin analogs. And rapamycin is one of those magic molecules that has been around for a long, long time. It has a liability, and people have not been able to really get rid of the liability. But that is changing now. There are at least 2 companies, that have actually engineered out the liability, and those are going into the clinic next year. So we'll actually have some really short-term readouts of rapamycin effects. And I think that will be really, really interesting because people have been waiting for this a long time. And that's just one single molecule, but you can actually look from worms all the way through rodents and mammals, and you see that rapamycin at a low dose has a tremendous effect. So we'll try to sort this out, and I think that is one of the short-term molecules that will come online. The other thing are just general processes like autophagy or proteostasis in general, where we know that proteostasis goes down with age. It's just out of whack. We get a lot of debris and oxidized proteins that are hanging around. If we can just oil that engine again, it's a very simple thing to do. We believe it's going to be a complementary effect to whatever else you do specifically on targets, because you will help the cells to detoxify. You will help the cells to reduce oxidative stress. And I think there are several molecules that will be in the clinic. There are several autophagy inducers going into CNS. We have one company as well in that field. So I think in the next couple of years, you will see autophagy inducers or modulators, and you will get the first readouts in man. And I think those are some really short-term signals we'll see. And I think they will be very beneficial, either alone or again in combination with other very directed approaches.
Fantastic. Thanks, Jens.
Yeah,
Marco, is this something that would appeal to Fabian?
Yeah, I think autophagy is of course one, but generally most cells do still harbor their own kind of cleaning systems in a certain way. So if you can help just cleaning up what is going bad, so aggregated proteins or whatever type it is, and get that balance back to kind of more the healthy side of things. I think there is definitely some things that Forbion is interested in, and I set up with a colleague. We co-founded Factory, which is a gene therapy company specifically looking at factorized antibodies against aggregated proteins in the brain, which is also looking at ALS, Alzheimer's, Parkinson's, and age-related diseases. So there we actually try to clean up the brain and help the brain to rebalance, let's say. I think so. And then if I may quickly respond to Kristen and Rita, and also James' comments on like the quick fix, or the getting something relatively quick in the next with trials of maybe like 6 months or a year or something, which is overseeable by most of us, and definitely by investors, which typically do not look at 20 or 30 years ahead. But if you can, like very in detail, look at what you're doing, what the biomarkers are that are changing in those like faster trials, or maybe up to a year, like CRP, like cholesterol in the past, that we have validated kind of important biomarkers also for neuro, of course, neurofilament. Then you can use those biomarkers also for the longer run, and then you make that kind of that switch to more prevention, which is, again, it's another investment. It's long trials, but it's definitely something I would say a great start to look at.
We're moving into this decade of healthy aging, so it would be good for our panelists to have the opportunity to speculate a little bit on what benefits gerascience might bring to patients over the 10 years of this decade of healthy aging. So perhaps I'll go back to the start, James, and ask you to talk a little bit about what we can look forward to over the next 10 years.
Speculation about new technologies is always a minefield. And so I'm sure I can maybe create the disclaimer for everyone on the panel saying like, all right, let's drop the guard a little bit and talk a bit aspirationally, not necessarily this is what will definitely happen, tie me to a post if I'm wrong. And so where I think we're going is that there are two swim lanes that are charging ahead in the present day really aggressively, right? And we've touched on both of them in the conversation so far. The first is that we have all of these novel molecules, whether it's new variants of rapamycin, newly discovered molecules or mechanisms in aging, all of these different mechanisms that are charging forward to the novel drugs that will be safe and effective at treating an existing disease in the clinic. And I think that by the end of this the end of this decade, we're going to have the first few of those molecules, and I don't know if it's going to be 3 or 5 or 20, have either clinical proof of concept in humans, or even the first few approvals starting to come through for those specific diseases, not for use in prevention yet, because those trials just take too long. And then the second swim lane that's charging ahead is this biomarkers and how do you run a trial for prevention kind of push that is happening all over the world. And I think we're going to have the first 1 or 2, and I don't know if it's metformin or alpha ketoglutarate, which is a non-drug compound that's where human trials are starting in Singapore, or one of the other 3 or 4 molecules that you could run one of these prevention trials for today. But I think we're going to see some human data coming out of those first couple of human trials this decade, and that's going to create a framework to run better and faster trials in this broader longevity space towards the end of this decade. And we're going to really explode into next decade, into the 2030s, where all of a sudden all of the innovation that's been made in the lab in the last 5 years enters the clinic in the next 5 years, will be used for prevention in the first part of the 2030s. That's my prognostication. I don't know if the rest of the group would agree with that, but that's where I see this field going in a broad in broad strokes.
That's a very positive framework, James, and thanks very much for sharing that with us. Rita, perhaps to look to the CNS space specifically, what are your hopes and aspirations for the next 10 years?
I love the way James framed the two swim lanes, and I think I'm going to use that analogy in the future and in other discussions. But one is, of course, I think we're going to see the readout in the next 3 to 5 years of some of the most impactful, potentially disease-modifying therapies for CNS diseases, including in ALS and in Parkinson's, and maybe on more of a little bit longer timeline, some of the really interesting drugs that are being tested in the clinic for subsets of Alzheimer's spectrum disease. So I think lots of information about the efficacy of those drugs, and you're going to see an explosion of approaches that build on from those. Completely agree with James about the explosion in biomarkers and how that could lead us to thinking about prevention in novel ways, not only from the point of view of clinical trials, but also I think the kind of distant horizon. If I could look out to the very end of the decade, I think we're going to see an explosion of approaches in the epidemiology of prevention. So lifestyle choices, that intersection, as I was saying before, between your genetics and the choices you make and the way you live your life, and how that affects the appearance and the development of diseases in populations, sort of precision medicine of aging that is currently lacking really good epidemiology to support it. So prevention being within our grasp, I think, as a concept, as a scientifically testable set of hypotheses by the end of the decade.
Fantastic. Thanks, Rita. Kristen, you're marching into the clinic with molecules now. What do you expect to deliver, and how do you expect to grow on the progress you've already made over the next 10 years?
Yeah, I think it's a really exciting decade for the field. I mean, I think that over the next 10 years, we and other companies on this call even, right, we'll probably try at least 10 different mechanisms of aging, like in human clinical trials. And that's never been true in the history of the field before. And honestly, 10 feels on the low side. Probably we can handle more than that, right? And I guess too, that I think that at least a couple of those are going to work, like not only for their initial indications, not only for improving biomarkers in the near term, but ultimately also to extend health span, to add years to healthy life. And I also think that for all of us, those first approvals for drugs that are really targeting mechanisms of aging are going to, again, sort of make the space grow even more, right? Like seeing that first sort of victory, I think, will help resource the space even more.
Fantastic. Thanks, Kristen. Marco, as an investor, what do you hope for your portfolio companies to deliver over the next 10 years?
Well, let's again use the analogy of James, but actually I'm a keen marathon runner, which typically goes a bit faster, but of course also needs some duration, durability. So I guess it's gonna take a lot of time, but a lot of money too. But I think that is available for great ideas, and definitely, well, Jens has actually a specific fund for healthy aging, and also Forbion is really onto that. So it's very exciting to be with those great scientists and co-founders of those companies. So an easy one, I think I could say, is in 10-20 years from now, we most likely live longer than we do now, with many more treatment options. And I would like to see, and I think we will see the biggest strides in neuro and CNS diseases. There is so much still unknown. With all the technologies we discussed today, with all the technologies and all the efforts that are ongoing, and all the clinical trials that are ongoing, and many will fail, we know, right? Many, especially in neuro, many will fail. But also from those failed trials, as long as we kind of combine that knowledge and understand what we are trying to do, I'm sure we'll make a great improvement. But it's also a hope, I would say, and not only an expectation. So I think that is an important one. And then, of course, it's not only age healthy, but also live healthy, right? So there's a lot of things we can do ourselves by not taking drugs and prevent diseases.
Thanks, Marco. Jens, I'd like to give you the chance to make a few closing comments yourself on, what you think the next 10 years will hold for this area of science.
Yeah, so I think I'll take it even broader. And I think this will be the decade where we see the paradigm shift from the way how medicine is done today, namely that you wait for a disease, you go to the doctor, and then you get a pill. I think we will see a future where you get a risk assessment somewhere in mid-age, you know your risk, and disease and medicine is more risk management than actually treatment of diseases. And I think that will completely change the way how we look at disease. And it will be ultra personalized. I think everybody will have the tools to actually go on a very personal health journey through their lives. And we will have tools, be it on measuring where your health is, to also actually early intervention, so that healthy aging and healthy living in old age becomes reality. So I think the next 10 years will be the beginning of that paradigm shift.
Jens, thanks very much. It seems like we have a lot of great impactful science to look forward to in this field. So on behalf of our audience and patients everywhere, I'd like to thank you all for your contributions to this increasingly important area of science. Rita, Kristen, James, Marco, and Jens, many thanks for joining us today, and our best wishes for your continued success. I look forward to discussing progress with all your ventures as we move through this decade of healthy aging. Thank you very much.
Thank you.
Thank you.
Thanks, Dave.
Thank you.